Don't you sometimes wish you had a twin? Someone who could clean your room for you, take your bio test, or just do all the things you don't want to do? Well, you can't. The reasons for that are not important, but if you really want an identical you, don't look at science experiments or laboratories, look at yourself, at your cells. As you go from a baby to a teen to an adult, it's not your cells that get bigger, there's just a whole lot more of them. In unicellular organisms, mitosis is used to make another organism. And in multicellular organisms like humans, it's used for cell growth, development, and cell repair. Mitosis itself is nuclear division, where the chromosomes divide, and cytokinesis is the physical splitting of the cytoplasm into two cells. During interphase, the cell appears to be sleeping, but it's really doing metabolic activity, getting ready for mitosis. In prophase, the nuclear membrane and the nucleolus begin to break down. The DNA inside condenses and forms chromosomes. The centrioles also start to move towards the poles. And as this is happening, the mitotic spindles, made from proteins called microtubules, begin to reach out and attach to the kinetochore. The kinetochores is where the sister chromatids are attached to each other. The chromosomes line up in the middle of the cell. The line the chromosomes form is called the metaphase plate. This is metaphase. From metaphase comes anaphase, and in this phase, the chromosomes that are lined up in the middle of the cell begin to separate at the kinetochores and move to the opposite ends of the cell. In telophase, the nucleoli and the nuclear envelope begin to reform, and the chromosomes begin to unwind. Cytokinesis is when the cytoplasm splits into two daughter cells. A cleavage furrow pinches through the cell until there are two. Or in plant cells, a cell plate forms. So there it is, the only way, admittedly, for humans to clone themselves. But you know, sometimes I still think it would be pretty cool if there were a lot more of me. 'Cause it would be. A lot more. The world would be a much better place with ponies, music, and mandatory television watching. But, as I've been reminded over and over again, one means more than enough. And besides, although I'm pretty awesome, my clones would be extremely sloppy, lazy, and terrible at all things physical. This is pretty much all the problems we would have if everything reproduced with mitosis. We would all be exact copies of each other, like a tween boy band. And this would mean that we would never be able to improve, and there would be no genetic diversity. This is why our sex cells, or gametes, undergo meiosis to reproduce. When our sex cells divide, they become haploid, or have half the number of chromosomes that a body cell does. Now, the sex cell shouldn't feel bad about itself. The reason it has half the number of chromosomes is that when the zygote is formed, it has to have a full set of chromosomes, half from the mother and half from the father, because it would be pretty weird if you were exactly like one of your parents. Now, you aren't an exact mix of both your parents either. If you were, your brothers and sisters would look just like you. To fix this, meiosis has a thing called crossing over and genetic recombination. Meiosis is divided into two phases, meiosis 1 and meiosis 2. Now, in the beginning of the whole process, you have interphase, which is very similar to the mitosis interphase in that you have DNA that is replicated so that each chromosome is made up of two sister chromatids. However, in meiosis, the chromosomes all have a partner, known as a homologous chromosome. These two chromosomes basically carry the same genetic information, but you get one from your mom and one from your dad. The four chromatids together are known as a tetrad. The first step of meiosis is prophase 1. In prophase 1, the nuclear envelope disappears, and so does the nucleoli, and the spindles begin to form. However, the major things that happen in prophase 1 are crossing over and genetic recombination, and you can thank these two processes for not looking like your siblings. So in crossing over, the double chromosomes line up, with homologous chromosomes from your mom and dad. These chromosomes get tangled up, which results in crossover. This leads to the two chromosomes swapping genetic information, which is recombination. The swapped information on the chromosomes are in the same location, so they are trading genes for the same trait. Like your mother's chromosomes might swap with your dad's for hair color. This creates genetic variation by having four chromatids that are all different, which is a key factor in natural selection. Now that all of that is done, next comes metaphase 1. In this step, the homologous chromosomes all line up along the metaphase plate. Then each individual chromosome is pulled apart and taken to opposite ends of the cell in anaphase 1. Then comes telophase 1, where the nuclear membrane and the nucleoli reform, and the cells split apart in cytokinesis. Now you have two cells that are both haploid, because keep in mind, though each chromosome is made up of two sister chromatids, they are all still just one chromosome. This means that the two daughter cells of meiosis 1 are haploid, having half the number of chromosomes of a somatic or body cell. That was complicated, but it isn't over yet. Next comes meiosis 2, which is basically like a crappy sequel that has the same story as the first movie and totally rips off another movie, in this case Mitosis. Sister, I think we've grown different. I feel like something has been pulling us apart. I think we're gonna have to go our own way, but you'll find someone who will make you feel whole. Goodbye. Goodbye, sister. So you have the two haploid cells, and then prophase 2 begins, and you basically know what happens. Nuclear envelope breaks, nucleoli disappears, DNA condenses, and so on. Then there's metaphase 2. The chromosomes line up along the metaphase plate. And in anaphase 2, each sister chromatid is pulled apart and taken to opposite sides of the cell. You still have the same number of chromosomes. The sister chromosomes have been split apart. Now comes telophase 2, where the nuclear envelope reforms, and then the two cells both divide with cytokinesis, and you are left with 4 haploid daughter cells that each have single chromatids. All of the 4 cells are different because each of the chromatids are different due to crossing over and recombination. And that is meiosis. Now when the sex cells, either a sperm or an egg, match up with the other one, you probably already know how that happens. They form a zygote. And that zygote will be diploid, having a full set of chromosomes, because 2 haploid cells are joined together. And that zygote can form into a baby. And this has been Bio Fun with Jordan. And I don't know how to make this exciting. Oh my god, that was my best line ever! You feel whole?
Exit. Other way. Keep doing it for a couple times. Stay. One more time. Jump up and down. Okay, that's good.
And this has been Bio Fun with Jordan. No, I don't like that.